Welcome to the TOEFL listening section. In this part, you will hear one or more conversations in an academic or campus context, such as a discussion between a student and a professor, or between two students. You may take notes while listening. Listen to part of a conversation. You will hear it only once, so listen carefully. After the conversation, you will answer questions about what you heard. Some questions will ask you about main ideas and details. Others will ask about implied meaning, speaker attitude, or how ideas relate to one another. Use both what is stated and what is implied to answer.
Good afternoon, Professor Morales. Thanks for meeting with me during office hours. I wanted to talk about my project on synthetic biology applications for industrial production.
Good afternoon, Alex. I am glad you came by. Tell me what angle you are planning to take. Synthetic biology is a broad field.
I am interested in engineering microbes to produce commodity chemicals and materials. I want to compare case studies and discuss technical challenges and biosafety issues.
That is a solid focus. In industry, synthetic biology is used to repurpose microbes for producing pharmaceuticals, biofuels, flavors, and bioplastics. Do you have specific examples in mind for your literature review?
Yes. I plan to discuss the engineered yeast that produces the precursor to artemisinin, microbial production of polyhydroxyalkanoates as bioplastics, and cyanobacteria engineered to secrete isoprene. I also want to include cell-free systems for rapid prototyping.
Those are excellent choices. The artemisinin example demonstrates pathway reconstruction in Saccharomyces cerevisiae to replace plant extraction. PHAs show microbial synthesis of polymers. Cyanobacteria highlight direct conversion of CO2 and light into hydrocarbons. And cell-free systems let you test pathways without maintaining living cells.
When we talk about engineering organisms, what are the usual steps companies take to go from a pathway idea to an industrial strain?
Typically, teams start with pathway design, using retrosynthesis and metabolic models. They choose a chassis organism suited to the product chemistry. Then they assemble and optimize genetic parts, use promoter tuning and ribosome binding site engineering, and apply directed evolution or adaptive laboratory evolution to improve enzymes and flux.
You mentioned chassis selection. How critical is choosing between E. coli, yeast, or non-traditional hosts like Yersinia lipolytica or Cupria vitis?
Chassis choice affects tolerance to solvents, secretion capabilities, native metabolic pathways, and regulatory compatibility. For lipids, Yersinia is attractive. For PHAs, Cupria vitis or engineered E. coli can work. For complex glycosylation and biologics, certain yeasts or mammalian cell lines are necessary.
What about biosafety and containment when deploying engineered organisms at scale? I know regulators care about that.
Biosafety is a major part of industrial application. Companies design genetic safeguards like kill switches, auxotrophy for synthetic amino acids, and multi-layer containment. They also use non-pathogenic chassis and physical containment in bioreactors. Documentation and environmental risk assessment are required by regulators.
Can you give me an example where a genetic safeguard was crucial in a real project?
One example is engineered bacteria intended for agricultural use that were modified to require a synthetic nutrient not found in nature. That way, the organism cannot survive outside supplied environments. Another example is kill switches that trigger cell death if specific environmental signals are detected.
What technical challenges do teams face when scaling from bench to industrial fermenters?
One key challenge is scaling lab successes to industrial fermenters without losing yield. Changes in oxygen transfer, mixing, shear stress, and gradient formation can alter metabolism. There are also challenges in enzyme stability, cofactor availability, and byproduct formation that become prominent at scale.
So yield often drops during scale-up. How do engineers mitigate that?
They use process development to adapt fermentation parameters, redesign strains for robustness, and implement fed batch strategies. Computational fluid dynamics can help model mixing. Sometimes teams move to continuous processes or use immobilized cells to reduce stress.
You mentioned cell-free systems earlier. How do those fit into industrial production?
Cell-free systems are powerful for prototyping and producing high-value molecules at smaller scales. They avoid cell membrane barriers and can perform reactions with optimized enzyme mixes. For large-scale commodity chemicals, they are not yet dominant, but they offer rapid design-build test cycles and can reduce time to market for certain products.
What role does computational design play in pathway optimization?
Computational tools predict pathway bottlenecks, flux distributions, and possible toxic intermediates. Genome-scale models help prioritize gene knockouts and overexpressions. Machine learning is also being applied to predict enzyme variants and to guide directed evolution experiments.
Are there economic or market considerations I should include in my paper?
Yes. Cost of feedstocks, downstream purification, regulatory approval timelines, and market price volatility all influence viability. For example, microbes producing rare flavors can fetch high prices, while commodity biofuels face stiff competition with petrochemicals unless policy incentives or carbon pricing shift the economics.
For methods and tools sections, I plan to describe CRISPR-based editing, synthetic promoters, metabolic flux analysis, and directed evolution. Does that cover the main technical toolkit?
That covers the essentials. Add modular cloning standards for reproducibility and examples of industrial analytics, like mass spectrometry for metabolite profiling. Also discuss regulatory frameworks and public perception, since social license affects deployment of engineered organisms.
That is helpful. For my case study section, I will compare artemisinin precursor production in yeast, PHA production in bacteria, and photosynthetic production of isoprene in cyanobacteria, and then analyze common bottlenecks.
That comparison will illustrate different engineering strategies and trade-offs. Make sure to highlight how each project handled enzyme selection, host optimization, scale-up, and biosafety.
I will. For next steps, I will draft an outline and send it to you. Should I include suggested experiments for a hypothetical industrial collaboration?
Yes, include a section with experimental plans and expected milestones. Also propose key performance indicators like titer, rate, and yield. Send me the outline, and we will refine the scope for a presentation.
Thank you, Professor Morales. This discussion clarified a lot and gave me concrete examples and analysis angles for synthetic biology applications.
You are welcome, Alex. I look forward to your outline and to seeing how you synthesize these case studies into a coherent assessment of industrial potential.
Listen again to part of the conversation. Then answer the question.
One key challenge is scaling lab successes to industrial fermenters without losing yield.
This concludes the conversation portion. For more TOEFL practice tests and study materials, visit Spinta.org.